That's how you walk in there. Okay, question in the front. Yeah.
What did you mean at purchasing the modules?
Oh yeah, so we're gonna actually start where we ended, and so ask me at the end, raise your hand if you still don't understand after we go through that. Okay. So quick business, for people still needing to make up the pre-test and or quiz 1, they'll be open this weekend. With quiz 2, those of you who are supposed to be taking it, I'll send you a password. Exam 1 is scheduled for the 24th. I'm considering moving it to the 20th because it looks like we might get there. So someone complained about me last semester, saying I was unorganized and moving exams all over the place. So I tried to explain it, that this is my first time teaching twice a week, so you have to be flexible. So if you're not going to be flexible, I'm sorry, it's going to be painful. Yeah.
24th is a Monday.
25th. Okay. 25th. I always think of week of, so sorry about that. Yes, 25th. So I'm thinking of moving it back to that Thursday. But I will let you know by the 11th, so you have plenty of time to adjust. I just don't want us to have to go into another topic and then test you on this, because that always sucks. Those interested in an oral exam for the first exam, please contact me a week in advance so that we could schedule it. You guys all have an option to make one exam an oral exam if you are a bad test taker and would rather sit in my office with me and chit-chat about science in place of an exam. That is a possibility. I say chit-chat, but really I am asking you to explain the same stuff that's on the exam. So just contact me if you're interested. A lot of people hear oral exam and get terrified, so don't contact me if you're terrified. No one has to do it. It's just an option. It's just an option. And if you're interested or you just want to explore the idea, sometimes what people do is they take the first exam and then see how they do and then start thinking about the oral exam. Tuesday, 12:30 and Thursday, 10 a.m. recitations are moving to SM 275. Hopefully that will make it a little more comfortable. The other ones, well, if you're in Sarah Texel's recitation, that stays the same. And the other one, I just couldn't move because there was no availability, so we just have to deal with it. I sent out some emails about that, so hopefully everyone gets it, and I'll make sure there's a sign on the door. So other recitations have not changed. So if you're not in either of those, you can turn off your ears. Now turn your ears back on. Okay, so last time we were talking about the concept of a cell as part of a larger organism that needs to respond to the environment. So that's the whole basis of this class. We talked about some methods, Northern, real-time PCR, microarrays, reporter assays. Someone says, do we need to know these methods? And I said, just understand them, why you would use them. Because it might come up on an exam question. Because I like to make exam questions kind of like experimental based. And so if I say, and you do real-time PCR and you find that this is upregulated by that, you should understand that what real-time PCR is. But I won't say what's the protocol for running a real-time PCR, because that's very uncool. That would be uncool. We start talking about chromatin structure, about nucleosome structure, post-translational modifications that are found on histones, and the protein domains that recognize them, the general function of post-translational modifications, and the general concept of protein domains. And so that's where we're going to start again. Okay, so I usually try to back up to like the topic we entered with, so the first slides will seem familiar. So protein domains, this is the part of a protein that does a certain job, which brings up your question about how they're modular. So I once tried to use an example of like modular prefabricated buildings, and no one liked that. So I'm not going to go there again. But what I want you to think about is Legos, okay? The big ones that like preschools have. So imagine you like take 3 different Legos and put them together to form one protein. But each of those 3 Legos has a different job. One Lego might have a bromodomain and therefore bind to acetylated lysines on histone tails. Another Lego might have an ATPase domain and therefore bind ATP and hydrolyze it and do some sort of work. And the other Lego might have, I don't know, some other job. Does that make sense? Okay, I once brought in Legos and did that. That was really fun. I just forgot to get them. So this is the concept of domains, right? So when I talk about how they're modular, there are these different bits that you can put together. So in reality, the domains have similar structure. So you can imagine taking one of those Legos and use it in 20 types of proteins that may have similar function in that little bit, in that little Lego of the larger Lego structure. And so this was this kind of example of 1, 2, 3. These are 4 different proteins, and this is just the domain structure. And they all have bromodomains. So you can make predictions about the function of a protein based on the domains that they have. And so you could say, okay, all of these proteins have the ability to bind to acetylated lysines on histone tails. This one has a helicase C domain. And so you can say, oh, what's that? Well, if you remember what helicases are, you would think, oh, it has something to do with unwinding DNA. If you don't know what it is, you're like, what's this NAT domain? Look it up. It's all out there. I think there's a whole database full of domains and what they all are. But it's useful just in terms of knowing the structure, not the structure, but the function of a protein. Okay, so this is where I realized, yes?
So different domains can be put together to perform, they all perform different things, but do they all still come together to perform the main thing?
Yeah, so you'll have a protein, and that protein may have a job of moving around nucleosomes so you can access promoters, right? But it'll have a domain that's involved with binding to acetylated lysine, so that's involved with recruitment of that protein to your nucleosome. It'll have a domain that maybe is involved in DNA unwinding, which will be involved in like the work that it does. It might have a domain that's involved in binding to ATP, and that's also helping with actually the movement of the protein. But it's still one function, and it just so happens there's not one protein in the genome that does each job. There's so many that do similar jobs. There might be 50 nucleosome remodeling complexes, which are made up of, which always have a protein in them that has a bromodomain. And it's just like, it's about efficiency. Like, this structure has evolved to function very well in binding to acetylated lysines, and it tends to be found over and over again in protein structures. Yes?
The relationship between functional units and protein domains?
The relationship between what?
Functional units.
Functional units and protein domains, the same thing. So I was trying to explain a domain as a functional unit. So each little Lego is a functional unit of the protein. It has a job that it does, and you can refer to it as a domain. Okay, any other questions about this? Okay, so this is where I realized that we were running late the other day, and I will never do that to you again, because that makes me late too. So what does this have to do? What does all this talk have to do with anything? Well, that's because the modification of histones, it affects the chromatin structure and its function. Structure affects function through increasing DNA accessibility and recruiting proteins that will affect structure and function. Okay, so these modifications kind of are your regulating side of everything. It's everything. Okay. Acetylation, methylation, phosphorylation, ubiquitination. All of these, and sometimes you'll have ubiquitination and acetylation competing for the same glycine, but all of these modifications occur often simultaneously, so it's not one or the other. Okay, so let's talk more specifically about these modifications. So histone acetylation is associated with active chromatin. So it's kind of activating, open kind of association, right? You'll have your closed chromatin. So your DNA is tightly wound around your histones. And then a histone acetyltransferase, or HAT, will come and acetylate histones, and that will result in the opening of the chromatin. So you can think that acetylation is associated with activation and gene expression, opening up the chromatin to expose binding sites for proteins that are involved in activating gene expression. And then histone deacetylation. What do you think deacetylation means? Okay. So often people get bogged down with these big words, and often the meaning is right in there, right? So it's removal of your acetyl group from the lysine and lysine, right? And so that's often associated with closing things up and silencing. And those are, so there's two enzymes involved here, one that's going to acetylate, one that's going to deacetylate. A HAT acetylates, a HDAC deacetylates. And it's right in there, right? Histone acetyltransferase, histone deacetylase, or, yeah. Okay? Okay. So I see a lot of people scribbling furiously. Is that stressing you out? Are you stressed out scribbling? Or you feel really good about furious scribbling? Okay. As long as you feel good about it. No stressing out. You can listen to it again. It's all saved forever. You can listen to it next semester if you want to. You can listen to it 2 years after you graduate. It's there. It's on YouTube. I'm sure at some point I'll take things down off of YouTube, but it's there. It's there. Okay. I don't want stressed out people in this class. Everyone needs to be happy and laid back and pretending you're like sitting on the beach somewhere. Huh?
Just give everybody a name. Don't take exams.
I know we were talking about that in recitation. I'm saying I wish I didn't have to give exams and I could just give everyone an A for being delightful people.
Yes.
I know,
That's an
I
awesome
wish I would be fired. And I'm sure Towson
But all
would
your
get a
students will be
really
happy, so.
bad reputation
We
for
won't tell.
selling
We
degrees.
won't kiss and tell. We don't do that.
Okay, let's get back to it. Sorry, sometimes when I see people like, I'm like, oh, are you okay? Okay. Okay, so let's talk about acetylation for a minute. So you know, you learn over and over again, acetylation, activation. Well, why? Right, so acetylation is actually removing the positive charge from lysine residues on core histone. So when you talk about acetylation of the histone core, so not the tails, but the core histone, or also it happens on the tails, but with the core histones, you actually, so here's your lysine. Does everyone remember their lysine structures? Let's just pretend. Here's your lysine, right over there. There's the structure. It has an amino, right? It has a positive. Remember your DNA has a negative charge. Everyone orient yourself to a picture for a minute. Right, positive, negative equals binding, right? Like magnets. Anyway, sorry. So when you have HAT activity and you acetylate the lysine, so here you're acting your acetyl group, and you're getting rid of that positive charge. So now you no longer have that tight binding with the DNA. Okay, and so remember I say acetylation is associated with opening up the DNA. So you can imagine if suddenly that tight interaction between the histones and the DNA loses that positive negative charge, you're going to be able to more easily open up the DNA. Okay? Kind of makes sense if you actually think about what's really happening. Yes?
And that happens when you have an acetylation, is that what you said?
This is acetylation. Yes, so this is about, so acetylation is adding an acetyl group to the lysine. And so that's this is actually, here's your normal lysine, so that's your deacetylated version, and here is your acetylated lysine over here. I have to get a better pointer, people always complain about it. How's the front row feel about my pointer? Can you see it?
Yeah.
Middle rows? Yeah, back row? Okay. Okay. So histones H3 and H4 are acetylated by histone acetyltransferase, and the acetyl groups are removed by HDAC. Okay. So there are a lot of enzymes that are involved in histone modification. Should you go memorize these? Don't memorize them. But understand there are different classes of enzymes. So if you want to think about acetylation, you should automatically think of HATs and HDACs. And so there's a bunch of them, but we don't need to remember, that there's TAF 250 and PCAF. No, because I don't remember that. There's a bunch of HDACs. There are whole families of proteins that are HDACs. Just understand acetylation, deacetylation, HAT, HDAC. Then you've got these histone methyltransferases. What are methyltransferases? What do you think they do? Yeah, they transfer methyl groups. It's right there in the name. Right, and then there are also histone demethylases. And then there are also DNA methyltransferases. What do you think DNA methyltransferases do? Transfer methyl groups to DNA. So it's right in the name. Don't get bogged down with long sounding things. Okay. Okay, so we're talking about acetylation and opening up the chromatin, and how if you acetylate on the core of your histone, you're going to loosen up the interaction with the DNA. You're also going to acetylate on histone tails. What is that doing? Well, it's creating a binding site. Remember, we talked all about post-translational modifications and how they change the shape of things and create binding sites for things. And so what acetylation is doing is creating a binding site for a bromodomain containing protein. Okay? And what is that bromodomain containing protein doing? It's recruiting other proteins. And with the main goal of activating transcription somehow. Okay? I'm being very broad, very general. These are not specific names of proteins with specific jobs, but it's more of a theme, right? Acetylate histone tails, recruit bromodomain containing proteins, recruit some proteins that activate something, gene expression in some way. Yeah?
Just to be clear, the bromodomains are on histone tails, right?
Bromodomains are on proteins that bind to acetylated lysines on histone tails. So bromodomains are, so let's go back to domains. Remember the Legos? So so this blue protein here is your multi-Lego protein, and it just so happens the Lego over here binds to acetylated lysine on the histone tail. Okay? People always get confused. Every year, I feel like I have to somehow learn how to explain it differently, because every year people get really confused about that. So if anyone has an easy way to explain this, come see me and teach me, because you guys could probably teach me how to explain it better. Okay, so then it recruits other proteins. And so here's just a picture of a bromodomain on a protein binding to acetylated lysine. Okay? Yes?
Does it happen in steps, or can a bromodomain protein be already attached to another lipid protein and then attach itself to acetylation?
Okay, so the question was, does it have to happen in a specific order, or can you have your blue protein already bound to your orange protein, and then it gets recruited? Absolutely, because it might be a whole protein complex where one protein in the complex of proteins has a bromodomain, right? And that's what's recruited. Really, the most important thing is there's a level of regulation. The level of regulation is about the acetylation. If there's no acetylation, can you recruit the protein? No. Today's like the one day I did not bring a drink of water. Okay, I'm gonna give you guys 1 minute to just think about this and discuss with each other while I go get a drink of water from my office before I talk about my line. Okay, chat for a minute. Talk about bromodomains. Where are they? What are they binding to? You have an extra one? Oh, it's okay. I'll be right back. You guys can have a
Okay, then what's happening over here? Is actually the protein. That's what I say, well, on the protein.
yes. Okay, so I just had a question. When the histone tail is acetylated, will a bromodomain protein always come? Well, when you create a binding site for something, it increases the probability of an interaction happening, right? So think about the cell as full of proteins, constantly moving around. And if there's a binding site, it's going to bind, right? So if the bromodomain containing protein is there, it'll bind. The question is it there? And so you might say to me, well, what determines that? Well, then you go back to gene expression. Is that protein expressed at a high enough level? Is it present? Right? So normally when you want to activate gene expression, you also have to upregulate expression of the genes involved in activating gene expression.
So most of the time the bromodomain will bind if it's there.
If it's there, it will bind. Absolutely. So this is your regulation. This is it. Okay, any questions? It's nice to have a break, right? Yeah.
So in order to separate histones from DNA, you acetylate the core, that the little drum thing, right? If you want to separate.
Go.
But if you want to bind like another protein, then you acetylate that tail.
Well, so it's not as simple as that. But so if you're acetylating the core, you're going to loosen the interaction. But if you acetylate the tail, you're going to recruit proteins that often are going to move your histones around. Yeah. So there's usually not going to be one or the other. Usually there's going to be multiple modifications happening in multiple places that are going to form like a histone code, which we're going to talk about in a minute. And it's going to have a meaning, and it will recruit a certain bunch of proteins that are going to do certain things. Okay? Yes?
Does anything else bind to acetylated tails of histones, or it's only bromodomain protein?
So there are many types of proteins that bind to acetylated lysine on histone tails. But most of them bind through having that bromodomain. Okay? So it's not one bromodomain protein, it's like 100 proteins, maybe with different functions, but all have a structure that's similar that interacts with the acetylated lysine. Okay? Questions up here?
So is the histone tail lysine itself?
No, so lysine is one amino acid,
Okay.
and the histone, remember, it's a protein chain, and it's made of amino acids. So there are lysines in that histone tail. Yeah. So it's just like this flexible piece of protein kind of hanging out of the acetylhistone. Okay, any other questions?
What regulates the enzymes that kind of initiate the acetylation?
So I just got the question, it's what regulates the HATs and HDACs, right? Well, so they need to be recruited. So usually what happens is you have some kind of gene regulatory protein that's going to combine to a sequence that's specific in the DNA and then recruit HATs or HDACs. Yeah. It's all regulated. And so that you'll say, well, what regulates that? Well, something's going to increase the expression of that protein. Okay. Any other questions? Good questions today. Okay. Let's talk about histone tail methylation. So histone tail methylation, just like acetylation, creates a binding site for recruitment of other proteins, except for the difference is these proteins have chromodomains which interact with the methylated lysine on the histone tail. Same concept, just a different point of regulation. So you can imagine sometimes the same lysine might be methylated or acetylated, and that's going to tell the cell what should be happening. Either you should be opening up your DNA, or you should be closing it up, you should be activating transcription, you should be silencing. So histone tail methylation is associated with heterochromatin formation. So methylation usually, not all the time, but most often is associated with silencing and heterochromatin formation. So our genes that are located in regions of the DNA that are bound by histones with methylated tails, are these genes expressed high or low? Or not at all? Low? Right, so when you're highly methylated, you're either in heterochromatin or you're silenced in some way. And so your gene expression for that gene in that region is going to be low or not at all. So again, enzymes that methylate, histone methyltransferase, right? Methyltransferase. You always know ACE is an enzyme, and so then you can just look at the rest of the word to figure out what it is. Enzymes that demethylate, histone demethylase. So you might think, well, these are a lot of enzymes we need to remember, but there's a way for you to get yourself to remember it just by understanding what it's saying. Any questions? Yes.
So DNA methylation expresses transcription.
DNA methylate represses.
But in this case, it's open. So
No, it's repressing.
repressing it.
It's repressing.
Okay.
Heterochromatin. Heterochromatin, silencing, repression, that's all associated with methylation. There are times that methylation has been shown with activation. I never like to say all the time everything, but usually if you hear methylation, you should think repression and silencing. You're right. Okay. And yep.
I'm sorry, so the repressing, it's repressing its function.
It's repressing gene expression. So anytime you have methylation of the DNA or of histones, it's usually involved with silencing or repression, repression of gene expression. Yep.
So does that mean if there's a activation,
The gene can be expressed, right, because it's opening everything.
Yeah.
So to close that, can you use the histone?
Yeah. Yeah.
Close that system?
Yeah, so usually when you see acetylation, you're going to have expression and opening of things, and methylation is more associated with closing of things and repression.
So in that case, you might use the deacetylation.
You're going to do both. So the question I just got, so what about deacetylation? Well, usually, so if you activate gene expression, you'll get acetylation. When that needs to be finished, you're going to get then deacetylation followed by methylation. Yeah, so it's not one or the other. It's all changing all the time. Well, depends, unless you're in very repressed chromatin. But anyways. Questions? I'm getting great questions. Everyone's thinking. Okay. So chromodomains are typically found in proteins that mediate transcriptional silencing. Makes sense because they bind to methylated histones. Right? Everyone remember that from about 2 seconds ago? Yep.
So how exactly does it silence gene? I mean,
How does it silence a gene? So it has to.
does it like not allow histones to open up the DNA, or what does it do?
So what do methylated? Okay. So it's involved with packing of histones into heterochromatin. So the question is, how does methylation silence gene expression? So one is it's involved with packing into heterochromatin. Remember, it's all about access. Do you have access to your promoter, essentially? That's what determines whether or not you're going to have gene expression. And so when you open things up, you're creating access. When you kind of fold everything on itself and tighten it and wind things tightly, you're silencing because you can't get any transcription factors in, you can't get your general transcription factors, you can't recruit RNA polymerase, none of that can happen because the binding sites are gone. Yeah.
So is it basically hiding the promoter region?
Yeah. So you can think of it as hiding the promoter region. Yeah. Yeah. Are we happy? Do you have a question?
So, basically, you have to first get histone acetylation before you can get a transcription factor bind. Or
So-
do you get transcription factor then histone?
Okay. So I just got, do you need acetylation and then a transcription factor, or do you need transcription factor then acetylation? So it really depends on the situation, right? And depends on what you're talking about. Sometimes you might have some kind of gene regulatory protein, like a transcription factor, that might bind to the DNA, but still the promoter is obscured, so you can't recruit your general transcription factors or your RNA polymerase. So that transcription factor might recruit a hat. Then the hat will acetylate the histone, tail the histone, and then the nucleosome remodeling complex will be brought in and move the nucleosome out of the way to open up that whole area, so then your general transcription factors and RNA polymerase can be recruited. Sometimes both are obscured, and then but usually you do kind of start the whole process with some sort of regulatory protein recruiting hats. Yeah. Okay. Any other questions? Yes. I had so many questions.
In acetylation, you talk about removing a positive charge and adding a negative charge. When methylation happens, is it adding an extra positive charge so it has a more attraction because it's repressing?
With methylation, are you adding more positive charge? Well, so don't forget with methylation, you have the multiple things happening. When you're having methylation of DNA, and sometimes you're having methylation of histones. So there's two separate things there. And then the other thing is one is like, yes, you're tightening up the binding, but also you're recruiting proteins that are helping in kind of spreading that silencing. So we're going to get into it a little bit more deeply. So maybe that'll clear things up. Okay, good. Sometimes when you start to talk, every year, when we start to talk about this, millions of questions come up. And I think it's because it's taking all the knowledge you already had and deepening it just enough to create 100 more questions, which is what science is. Okay, so this is what acetylation and methylation look like. First of all, remember, they're both on lysines. And sometimes you can get competition on the same lysine for both modifications. So here's acetylation. So right, your lysine, you have this amino group, and then here you get your acetylation. And then you can have mono, di, or trimethylation of your lysines, and that's another level of specificity because it's changing the shape of the protein actually differently each time, right? You can imagine this is a much larger shape than this one, right? So again, you're going to recruit different proteins. Okay. And then I put in phosphorylation because let's not forget phosphorylation does happen on histones and is involved with recruitment of different proteins, but we're not focusing too much on it. But again, often you have serine phosphorylation, so there's your serine structure, and that's just adding a phosphate group. So there's all these modifications we're talking about, right? And they occur on the core as well as histone tails, and there's many happening at the same time. So this is just showing, for example, all the different types of modifications that you can find. Okay. In addition to that, you've got phosphorylation, ubiquitination, sumylation. They all regulate different processes, also by creating binding sites. For example, phosphorylation is involved in expression of genes involved in cell growth as well as chromosome compensation. And then ubiquitination and sumylation are associated with transcription and DNA repair. And so again, you're recruiting different proteins to different sites in the genome that are going to be involved with activating different functions. Okay? Why did I put that in? That's kind of redundant. Okay, so now I'm saying, right, there's all these different modifications happening. So you can imagine the histone tails are going to have all these different shapes to them depending on which modifications are present and recruit different proteins. And so this kind of introduces the concept of the histone code, which I've referred to already a little bit, right? So they're all kind of forming this code that's recognized by a code reader, which is really a complex of proteins that will bind to those modified histone tails and recruit other proteins to do whatever's supposed to do. So you can imagine it like each code has an associated function. So each code is going to recruit the proteins that are needed to perform that associated function. So for example, my DNA repair is one of my favorite ones. And so you're going to recruit, so if you have all these modifications somewhere close to damaged DNA, then you're going to have recruitment of a lot of DNA repair proteins. If you're trying to silence this region of the genome in heterochromatin, you're going to recruit a different class of proteins, right? Because the same proteins aren't going to be involved in both functions. So there's this code reader, and so that's kind of shown here. And it's really this multi-protein complex with many different chromodomains or bromodomains or whatever that's going to recognize the different modifications and bind and recruit other proteins. Okay? So what do I mean by histone code? So for example, if you have methylation on lysine 9, then you'll get heterochromatin formation and gene silencing. But if you have acetylation on lysine 9, but methylation on lysine 4, remember I said methylation is not always silencing, you actually get gene expression. And if you have phosphorylation on serine 10 and acetylation on lysine 14, you might get gene expression. And if you get methylation on lysine 27, that's involved in exon activation. So this is the code, and it's all about recruitment of other proteins to do whatever needs to be done. Okay? So how do you think this works? Yep.
So these modifications can happen on other amino acids as well.
Not really. So lysines. So the question was, can these modifications happen on other amino acids? Methylation and acetylation tend to happen on lysines. Phosphorylation can happen on a number of amino acids. Here we're talking about serine, but there can also be threonine. What else? Tyrosine, thank you, I'm like trying to remember. Yeah, so phosphorylation's a little bit more. Also, same with stimulation and ubiquitination, only lysine. Whatever reason, lysine. Yeah, so what do you think? So how is this happening? So I'm telling you there's this code and it's associated, I also have a meeting and it's associated with different outcomes. How does this code, how is this code read and how does it result in multiple outcomes? What do you think is happening here? Yeah.
I'm assuming it's probably changing the shape of the tail and the proteins is going to bind specifically.
Right, so it's changing the shape of the tail, so it's recruiting a different code reader, right? That's going to bind to different shapes, and that code reader, what's that code reader going to do? Recruit the correct protein, recruit different proteins. Yep, what were you going to say? Recruit other proteins that are specific to the function. So if it's repression, it will recruit proteins associated with repression. If it's gene expression, it'll recruit proteins associated with gene expression. Questions? Okay. Right, so histone code readers might recruit other proteins, such as writers. You can imagine, so this is not scientific at all. This is Bruce Alberts and his minions' way of, well, Bruce Albert, you know he didn't write that whole book, right? You know he had help. Okay, anyways, I don't know. Bruce Alberts, oh my God, my career's over. Sorry, Bruce Alberts comes up with this method of explaining this. I know, I'm dead, right? I'm done, I'll never be funded. Sorry, love Bruce Alberts. Actually, it is my favorite book. But this is not like, you know, if you go to like a molecular biology conference and you're like, well, I work on the reader-writer complex, people will be like, you clearly read molecular biology of the cell. But it is kind of a nice way to think about it because the readers are proteins that are kind of reading the code, and the writers are the ones that are writing the code. And so in this case, it's explaining how you're going to spread that code to different histones along the chromatin. And so in this case, you know, there's a gene regulatory protein that's recruited to some sequence in the DNA, and it's going to recruit some kind of writer. So that's the thing where, you know, some protein recruits the first hat, and then the hat acetylates something. And then the code reader is going to come in and read that and say, hey, I need to recruit another hat. And so it's going to recruit another hat, which is then going to acetylate the next histone, and all along the chromatin until you don't need to be silenced anymore. Okay. So if heterochromatin is DNA that's tightly packed into chromatin and not available for transcription, what needs to happen for transcription to occur? Well, yeah, but more, okay, we got real specific. But more specifically, what needs to happen so that transcription can occur? Yeah. Okay, so a hat needs to come in and acetylate and promote. So more generally, you need to expose your promoter region, and so sometimes that is associated with acetylation. What else? Did I see someone else? Were you raising your hand? No, you. So transcription factors are going to be involved in that. But to expose your promoter or some regulatory region of your DNA that's tightly packed, what needs to happen to those histones? They need to be moved. Right? There needs to be some remodeling, right? Yes?
So could that be done using like histone deacetylase?
So histone deacetylase is associated with silencing. But if you need to activate, yes, you will need to do multiple things, but one of them will be acetylation. So that will be hat activity. What did I hear over here? Helicase. Oh. But actually, so keep that in your mind there, because we're about to talk about nucleosome remodeling complexes, which tend to have a domain that has helicase activity. Yeah. Yes. Anyone else? Look, someone around me is wearing something lavendery. It's not lavender. Yeah, it's like I'm in a French garden.
When you're doing that promoter, some part, enhancer part of the gear is like the top of the gear is like flips up, and that's the binder complex, right?
Yep.
To-
Yep, and we're gonna get there. So you can imagine, so I just had someone say in the front here, that, oh wait, so you usually have there's an enhancer region and there's a promoter region and it kind of like folds over. That all has to happen, and so that's like a very large region of DNA that needs to be open, right, for that to happen. So, and when we talk specifically about transcription, we'll see in gory detail how that all works. But yeah, usually you have this enhancer that's going to bind your transcription factor, or your transcription factor is going to bind your enhancer, and that's going to recruit all your general transcription factors to your promoter, and then recruit your RNA polymerase. Okay, so we're talking about what's required to activate gene expression when you're kind of repressed, and so I heard about HACs, and we're talking about nucleosome remodeling complexes. So sometimes I'll say nucleosome remodeling, and sometimes I'll say chromatin remodeling. I mean the same thing. Really, what I'm recovering from is using two different textbooks that have different language. So just know that they're the same thing. So chromatin remodeling alters the location of nucleosomes. And it's exactly what it says, right? You're remodeling your chromatin. It's like remodeling your kitchen. You might move your sink from this wall to this wall. Or just moving the nucleosome, not a sink. I love it. Every now and then I catch someone going, like, what are you talking about? I was just having a memory of my own kitchen remodel. We bought this house when I was in grad school. I moved to Baltimore from Boston. In Boston, it's like, oh God, like 2-bedroom condos were going for like $350,000. We never had a prayer of ever owning anything in Boston. So when we moved to Baltimore in 2002, Baltimore City, in 2002, it was like, oh my God, let's buy a house right away. So we bought this house that was like, the kitchen was horrible. And I remember we had this real estate agent who, it turns out, I don't think he stayed a real estate agent very long. He was new. He used to be a carny, like working carnivals. Like he actually told us about this. And so he was changing his career. Very nice, but like we walked into these houses and he was like, so do you guys eat? Like, do you guys cook? Are you looking for, I'm like, of course we cook, but apparently people out there don't cook. And so he kept showing us these houses with these horrible kitchens, one of which we bought. And then we had to remodel the kitchen. That's what wedding presents are for. Anyways, I digress. We're remodeling our chromatin here because we need to move our nucleosomes around. Remembering, demo was fun. Do your own demo. Okay. Sledgehammer, kitchen cabinets, good stuff. Right. So in this figure, what you're showing is you've got, in this case, you can imagine this first pink line might be some enhancer region, and maybe the second pink line is your promoter region, and it's wrapped around your histone. So there's no way that anything can bind to it because it's protected, it's busy, it's not available. And what you need is access. You need them to be in a place where something can bind to them, and which is, here it's showing these lime green proteins. So you need to move your nucleosomes. So don't think of histones as like this is where they are, this is where they stay. They're dynamic, and they need to move. So you can expose transcription factor binding sites, promoter and enhancer regions. So basically, it's so dynamic that you'll have things moving within milliseconds. And that allows enough time, that'll open things up for a long enough time for a DNA binding protein to be able to come in and bind to that region. So nucleosomes are actually moved around on the DNA by ATP-dependent nucleosome/chromatin remodeling complexes. So they require ATP. Yeah.
When histones are remodeling, is it just taking when it's exposing, I guess, the binding site, is it moving along the DNA, so a portion of DNA that was naked is now part of the nucleosome?
Yeah, so I need to look more deeply into this, but according to the textbook, which is always a few years behind, it was saying that sometimes you can think of it as the nucleosome. So the question was about whether or not it's just sliding the nucleosome along, and so an exposed region of DNA is now wound up around a histone. And so I feel like maybe it's not 100%, or maybe a combination of both, where sometimes you're just sliding things down, and sometimes you're actually physically removing the histone. And you can imagine sometimes you need that, right? I don't know how you're going to do transcription unless you have like a very large region that's free of histones, right? Or at least things are slid away enough for RNA polymerase to come through. So nucleosome remodeling complexes move around histones. Yes.
Okay, so once it slides on the first histone, is there something to stop it, to prevent it from winding itself to the next nucleosome?
So you said once that region of DNA
It's exposed.
is
like
exposed,
is there something that stops it?
so you're saying is there something that stops it from another nucleosome coming?
No, is there something that stops it to prevent it from winding itself to the next nucleosome down?
So think of it this way. So kind of like what we spoke about in replication is you're kind of limited in how much DNA is going to wrap around each nucleosome. So it's not like it's going to just keep winding around the next one. So only about 200 base pairs or so can wrap around each nucleosome. So I'm guessing that, if you slide it out of the way, it's kind of like limited in how much can be wrapped around. So if you slide them all over, think of like the beads on a string, kind of slide all your beads out of the way. It's just going to be out of the way. There are sequences in the DNA that help to control how far heterochromatin spreads. So, but that's kind of different. Yeah. Did I answer you, or are you unsatisfied? Okay, well, I think we need to clarify the question for me.
I
Okay.
think were you asking why is it 200 base pair?
No,
Why not?
like we have these two bottles, and you have the wire, the DNA
Yeah.
coming down off of this one, and let's say my promoter's right here. Is there something to keep it here, or is it gonna come down and wind itself to the next?
Oh, so think of it not as the DNA moving, but think of it as the nucleosome moving. The histones moving. Does that make sense? Okay. So I think we're thinking of it as like your DNA is moving around, but think of it as your histones being moved. Okay. I think that clarifies things a little. Okay. That was good. Use the props that are around. Okay, did everyone catch that? I know sometimes in the back it's hard. This class tends to kind of all happen like right here, and I really try hard to not exclude the rest of you. But feel free to come closer if you feel excluded. I love having friends. Okay. But please let me know if you don't catch anything. Yes?
What you think is that it's not like the DNA moving,
It's the histone moving, not the DNA moving. Yeah. 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 So these chromatin remodeling complexes actually are interacting with the histones and the DNA, but helping to either slide them down along the DNA or remove them completely. Yeah. Are we happy? Okay. I like it when people nod. Okay, so these nucleosome remodeling complexes are usually multi-subunit enzyme complexes. They're moving these nucleosomes along the DNA or removing them from the DNA. And so here's just the domain organization of 4 different ones, and you can look and see that they have similar domains. So for example,
This orange is this HeLa-C, which is it's an ATPase domain, and then you've got these promo and bromo domains. That kind of makes sense. Why you might have promo domains and bromo domains in nucleosome remodeling complexes, right? Who can tell me why you might have a chromo domain or a bromo domain in a nucleosome remodeling complex? Do you guys want to take a minute to talk to your friends about it? Do you need a break? Sometimes it's nice to take a break and be like, what was she talking about? Or should we just power through it? Yep.
Is one of them using the destruction of
But give any more detail than that.
the acetylation?
Right. Right. So I just heard acetylation and methylation. Right. So remember, all this acetylation and methylation has a purpose, and it's recruiting proteins. So you can imagine that, for example, this SWI/SNF family of nucleosome remodeling complexes that has a bromo domain, it's most likely to be associated with what? Acetylation and activation of gene expression. Because you're going to move nucleosomes sometimes to move them out of the way, sometimes to move them back, sometimes to activate gene expression, sometimes to silence, right? And so you've got bromo domains, and then here you've got these chromo domains. So there, those chromo domain containing nucleosome remodeling complexes are more apt to be involved in silencing. So this is kind of how we can predict the function of a protein, just by looking at the conserved domains that they contain. And then they all have, a lot of them have these, well, some of them, I should say, have these SANT-SLIDE domains, and they are involved with binding to histone tails. Again, it's logical, right? Because they are going to move nucleosomes, right? Okay, any questions about this? So I know a lot of you are probably not used to looking at the domain organization of a protein. It's usually shown just by making this line and kind of like denoting which region of the protein. So you can imagine your N terminus is here and your C terminus is here. And you know, if you looked really closely, you would have an amino acid sequence here, and just these amino acids create the bromo domain. These amino acids create the HeLa-C domain, and so on.
Yeah, I'm having a hard time for some reason visualizing like the histone tails versus like the naked DNA that's stringing them together. So they're separate, right?
Yeah, I had a figure, I think it was last week. No, maybe just on Monday. Let me just draw real quick. So the question was about visualizing the histone tails versus the DNA that's wrapped around the histones. So that would be your histone. That's really small. Okay, better? And remember, it's got 4 subunits. And then you're going to have your DNA wrapped around it, right? Okay, so there's your DNA wrapped around your histone. But remember, each of these, you have your H2A, H2B, all that. Each of these has their own protein structure, right? And it has an N terminus and a C terminus. Remember, that's your beginning and your end of your protein. And so it just so happens your ends are kind of hanging out here. And so you can imagine, I'm drawing sideways. And so these create nice little landing pads for proteins, right? So you can imagine if this one is acetylated, then that changes the shape there, right? And so you can get recruitment of some kind of protein that's going to land right here. Okay? Does that help people at all? And so the DNA is over here. It's neither here nor there. You don't even have to pay attention to it in this case. Yeah? Does the C terminus also hang out in the
No, I believe it's only the N terminal.
Yep.
Question is that where's the C terminus? It's folded up in there. Yep. There was if you go back somewhere
in the lecture from either Tuesday or last Thursday, I have a nice picture of the assembly of the histone that shows the tails hanging out. And it's in the textbook too, I think. Nope, that's not what I wanted. Let's try that. Okay. Okay. So nucleosome remodeling complexes, along with another protein called histone chaperones, they alter the position of nucleosomes on the DNA. And so they either sliding things along or removing histones and assembling them elsewhere. So you can remember these histone chaperones, they're chaperones. Chaperones tend to chaperone things, right? Like if you're a chaperone at a dance, you're usually watching some young children. Something like that. So they're histone chaperones. And so you're either sliding things around or you're removing them. Sometimes what you might do is you're going to remove a histone dimer and replace it with a histone variant, which we didn't talk about too much yet. Histone variants look just like histones, but they signal other processes. So these are your histone variants. And they have different functions. So you can imagine, as with everything in cell molecular biology, it all has to do with which protein is being recruited and the job of the protein that's being recruited. And there's some sort of regulation that controls whether or not that protein is going to be recruited or not. Right, so there's H3, H3.3, SEMP-A, H2A, H2AX. Don't memorize this, please. My favorite one, I have two favorite ones, actually. One is SEMP-A, and it's involved in centromere function and kinetochore assembly. What's the centromere? Does anyone remember that? Right, middle part of your chromosome. And what's the kinetochore? Attaches. Sister chromatin, right? So you have your centromere, and your kinetochore sets up there, and that binds your mitotic spindles involved with chromosome segregation during mitosis. And so in that region of the DNA, you have, instead of your normal histones, you have SEMP-A kind of taking place of one of the dimers. So you can imagine SEMP-A has something to do with setting up the kinetochore and interaction with mitotic spindle and all of that process. Another favorite of mine is H2AX, which is involved in DNA repair. And at the end of the semester, we do a little bit on DNA damage and repair. And I guess those of you who took cancer bio, who took cancer bio? Did you guys learn about DNA damage and repair at all, or no? A little bit? A lot?
A lot.
Oh, good. So I don't have to teach it. It's always at the end, so I often have to kind of like fly through it. So at least someone's going to know something about it. Yeah, so H2AX is a histone variant that's added in areas of DNA damage and is involved in recruitment of DNA repair proteins. Okay, so I think we're going to end with a little practice problem so that you guys can stop listening to me talk because I'm tired of it. So this is a histone variant practice problem. You're working at a pharmaceutical company because you graduated from Towson with a degree in biology and you got a job. Yay! Jobs are great. Okay. And you're trying to identify new chemotherapy for cancer treatment. So you're screening drugs, tons and tons of drugs. And you know what you're looking for because a good cancer drug should cause cell death. And one way of causing cell death is by causing DNA damage. It's counterintuitive, I know. But actually causing DNA damage will activate apoptosis and cell cycle arrest, and that's good because you want to kill those cancer cells, right? So a good drug will cause DNA damage. And you know H2AX is associated with DNA damage and repair. And then you also know that CHEK2 is recruited to DNA damage through H2AX. So what you decide to do is you're treating with all these different compounds, and you're screening through immunofluorescence, which is like no one would ever screen drugs this way, but this is a practice problem. You're screening with immunofluorescence. So quickly, I'm just going to tell you about how this looks. So the blue is DAPI, and so these are the same cell with different pictures taken using different filters to see different things. So in this case, it's DAPI, which stains DNA. Here, this is an antibody against CHEK2, which is this protein that's recruited to sites of DNA damage. This is an antibody against H2AX, again, this histone variant that's associated with DNA damage. And then this is a merge where you overlap them all, and that's typically how you look at immunofluorescence. And here it's colored, so it looks pretty. And so you're screening for cells that are treated with different cancer drugs. So my question for you is compound X, because this is cells treated with compound X, is it a good cancer drug? So why don't you take 1 minute? It might be obvious to some, others might not know what the hell's going on. So take a minute to digest, to think, to chat with a friend, and then we'll discuss. I'll circulate, and you can tell me over.
Now, we'll do the same thing when we all you and me right on.
Not
Yeah. So, okay, so I had a few comments and then I'll let you guys go. So someone said to me, well, one person was like, I don't understand what I'm looking at, which is a really good question. Comment, statement, whatever. So DAPI means that it's a stain that's staining the DNA. CHEK2, phosphotyrosine 26, means that it's an antibody that's binding to CHEK2. H2 gamma H2X means it's an antibody binding to gamma H2X. So it's a way to look to see if something's there or not. If you see dots, then the antibody's binding, and you can see. Use a secondary antibody that helps to give it some color. So that's one thing. The other thing is someone said, but how do you know if this is enough damage to be a good cancer drug? And I'm like, well, you're right. A properly controlled experiment should have an untreated control. And that would be another picture where you treated your cells with nothing and stained for the same things. And what you would see is this should be black, there should be nothing. Because gamma H2AX is a histone variant that is only added into histones at sites of damage. And then gamma H2AX recruits CHEK2. So then I had another student who said, oh, well, there's overlapping, like you can see the same exact dots for both proteins. And that's exactly the function of H2AX, is it binds where the damage is and recruits CHEK2, which is involved in activating repair. So that's exactly what should be happening. And another comment that was about, well, how can we fix this? And no, we're not trying to fix anything. It's just kind of a screen for whether or not we're causing damage. Because if you can cause damage, you can kill a cell. And since we're trying to find a drug to kill cancer cells, we're trying to cause damage. And with that, I'll let you guys go. We can start with this on Tuesday.
Answer it.
Oh, it's a good drug.